Hi, I am Simona Soverini from the University of Bologna in Italy. Here at SOHO 2016, I'm gonna talk about the perspectives and future directions for hematological oncology research. So we have been living very exciting times in hematological oncology due to the ever-increasing number of new technologies and research tools and the ever-increasing number of novel promising therapeutic approaches. As far as novel technologies and research tools are concerned, I would like to tell you a little bit about the new genome editing technologies that are revolutionizing the way we do research and maybe in the future also the way we treat some diseases. The CRISPR-Cas9 genome editing technology is borrowed from bacteria, where it serves as adaptive immune strategy that uses RNA-guided nucleases to cleave foreign genetic elements like plasmids and phages. It consists of the Cas9 nuclease that introduces DNA double-strand breaks and whose target specificity is dictated by base pairing between a short guide RNA and a DNA to be cut. So we just need to design a guide RNA complementary to the DNA region we wish to modify. The double-strand breaks can be repaired by non-homologous hand joining or by homologous directed recombination. Non-homologous hand joining is highly error-prone. It involves direct linking of the broken ends, and insertion or loss of nucleotides is a frequent event, resulting in disruptive indels. Homologous directed repair has much greater fidelity, but requires a DNA template to repair the lesion. By supplying an appropriately designed exogenous DNA, we may thus introduce point mutations or genomic pieces like promoters or entire genes. Thus, among the many applications of CRISPR-Cas9, we can have generation of indels to model loss of function mutation, introduction of point mutations to model gain of function mutation, insertion or deletion of entire genes, and generation of chromosomal rearrangements. More recently, non-editing applications have also been described that use a nuclease-dead version of Cas9 conjugated with a transcriptional regulator to turn on or turn off gene, or with an epigenetic modifier to introduce chromatin modifications, or with a fluorescent reporter to perform in vitro imaging of the cellular genome. Advantages and current and future applications of the CRISPR-Cas9 technology include the possibility to perform multiplex editing for a more accurate modeling of complex diseases, easier and more rapid generation of cellular and animal models, easier functional genomic screens, live imaging of the cellular genome. There's a broad range of future therapeutic applications, including, but not limited to, manipulating the epigenome, improving CAR T-cell therapy, and repairing disease or cancer-causing mutations, gene therapy. But long-term, physiological effects and safety are unclear, so using humans is not exactly behind the corner. As far as novel therapeutic strategies are concerned, I would like to talk about immune checkpoint inhibitors. Checkpoint inhibitors relieve suppressive signals exerted by cancer cells on host T cells to unleash anti-tumor T-cell activity. Checkpoint inhibitors were first tested in melanoma and other solid tumors, but they harbor great potential in several forms of hematologic malignancies, where PD-L1 and PD-L2 are frequently overexpressed via genetic alterations, activation of JAK-STAT signaling pathway, or viral infection, like EBV in Hodgkin lymphomas. There are several avenues of clinical research we need to embark on to optimize the use of immune checkpoint inhibitors in hematology. First, which hematologic malignancies to target? Promising results have been obtained in lymphoid malignancies, but there is evidence that also myeloid malignancies like MDS and AML might benefit from them. Which immune checkpoint to target? Besides PD-1 and CTLA-4, there are many more stimulatory and inhibitory co-receptors that modulate the intensity and duration of T-cell responses. and monoclonal antibodies against some of them are or will be in the clinical trials soon. Which combinatorial approaches might be worth testing with cytotoxic agents, with different checkpoint agents, other types of immunotherapy, and so on? How to select patients for treatment, because solid biomarkers of responses are needed, and finally, how to counteract resistance. In this regard, I would like to focus your attention on the manipulation of the gut microbiota to promote immune-mediated tumor control. An interesting new concept is that the individual microbiome influences the immune fitness, promoting and calibrating both innate and adaptive immunity. Determining the precise microbiome of a given patient is now becoming possible with the development of metagenomics, metatranscriptomics, and culturomics platforms. Information from these techniques can be used to direct microbiota conditioning strategies aimed to enhance the immune fitness. Another interesting avenue of research aimed to enhance the efficacy of immunotherapies is counteracting age-related change in the immune system, or immunosenescence. Hematopoietic stem cell aging is considered the main underlying cause of immunosenescence. Stem cell aging has long been considered to be irreversible until recent findings in animal models have indicated that several molecular pathways might be targeted to rejuvenate hematopoietic stem cells, hence to reverse some aspects of immunosenescence. Reversal of the global epigenetic profiles of aged hematopoietic stem cells and inhibition of mTOR and CDC42 pathways have been shown to be promising approaches. In conclusion, novel exciting technologies and research tools are becoming available to further empower the way we may study the biology of hematologic malignancies and develop novel therapeutic approaches. Functional ratification of the genetic alteration identified in omics studies will be greatly streamlined by CRISPR-Cas9 genome editing tools and their recent modifications. Whether and when CRISPR-Cas9 will have real therapeutic applications is early to tell. We are still at the dawn of the use of immune checkpoint inhibitors in leukemia. Careful correlative studies conducted in the context of large clinical trials are needed to shed light on several issues that may be critical for optimal use of immune checkpoint inhibitors. And modulation of immune fitness and the reversal of immune senescence are the most intriguing among these issues. Thank you for your attention.